In part two, we left off by talking about the general phases and the general events that are going to occur in our muscle contraction cycle. So we talked about what happens at the neuromuscular junction, what happens along the sarcolemma and T-tubule, and what's going to happen at the level of the sarcomere, right? So this part, we're going to focus in a bit more on some of those specific events and those specific things that happen at each of these different phases, all right? So first, at the neuromuscular junction, this is what we kind of alluded to it also at the end of the previous section or the previous part, is that we're going to have the action potential arrive at the synaptic knob or the synaptic terminal, right? So in chapter 8, the lecture before this one, we talked a lot about the action potential, okay? And so what this is, this first part here, is essentially just that. It's the action potential and it arriving at the neuromuscular junction, arriving at this region called the motor end plate of the muscle fiber. And so if you don't understand the action potential, I urge you to watch that first and to understand that first, and then move on and progress into talking about the skeletal muscle steps that we're talking about here. All right. So first, when you have this action potential arrive, it stimulates what are called voltage-gated calcium channels in the synaptic knob. So in this region here, it stimulates them to open. So calcium opens, and you have an influx of calcium from the extracellular fluid to the intracellular environment of the neuron, right? It turns out that calcium is the stimulus that is needed for the synaptic vesicles that have their acetylcholine. That's the stimulus for them to begin releasing and exocytosing their neurotransmitters into the synaptic cleft, right? So we begin releasing acetylcholine into the synaptic cleft upon calcium influx into the cytoplasm of the neuron. And then that acetylcholine, once it's in its uh, extracellular environment, once it is in the synaptic cleft and it has bound to its receptor, it causes that receptor to open, right? So this receptor, is it voltage gated or is it ligand gated? Well, it turns out that it's ligand gated because it's binding a ligand. It's binding some molecule, and in this case, the molecule is acetylcholine. So this ligand-gated receptor, it also functions as a channel. So after it binds its ligand, it binds acetylcholine, it then opens, and when it opens, it allows sodium to rush into the cell that we'll talk about in just a second. So here you can see calcium rushing in, vesicles beginning to fuse and exocytose their neurotransmitter into the synaptic cleft, and the neurotransmitter binding to its ligand-gated receptor or ligand-gated channel, binds it, it opens, and then it allows sodium to begin rushing in. So if you have access to the slides, you can click this video and you can watch the impulse travel down the neuron to the muscle fiber. So once we have done that, so we've bound acetylcholine to our motor end plate, and this is the neuron, this is the muscle fiber. This region here is known as the motor end plate. All right. So what we have first is you have something called the end plate potential, right? And this is essentially this occurs after we bind acetylcholine to the plate. You begin to have an influx of sodium into the sarcoplasm, so into this intracellular environment of the muscle cell, and this causes the membrane potential of the um, muscle fiber to begin increasing, right? So we're essentially getting it up to threshold, if you will. So we begin moving it up, and the resting membrane potential of the muscle fiber is not exactly the same as that of the neuron. It's a little more negative, so it's around -90, whereas the neuron was about -70 or so. The specifics are not overly important, but if you know, we're doing the same thing essentially. We have sodium coming in, we're getting our membrane up to threshold, and then once we're at threshold, you then have the opening of voltage-gated sodium channels adjacent to these ligand-gated channels. And then you have more sodium rush in. Sodium rushes in, causes the depolarization of the membrane, right? So that's where we're at here. We have sodium, we reach threshold, sodium rushes into the cell. We begin depolarizing, the membrane potential is becoming more and more positive. We then reach some peak, those sodium channels begin to close, and then we have the opening of potassium channels. Potassium channels open, allow potassium to rush out of the cell or diffuse out of the cell, and this causes the repolarization of the membrane, right? So we start bringing the membrane potential back down, more and more negative just as we did with our neuron, right? So it's really important, if you understand the basis of the neuronal action potential, then the events that are happening here along the sarcolemma of the motor end plate or of the muscle fiber, it's very easy to understand if you already understand the action potential. So again, if you don't understand it, I urge you to go back and watch that first, and then move on and start looking at this. Okay, so we take this end plate potential, and then we have this action potential, and it's traveling along the sarcolemma here, right? So we said earlier that there are areas where the sarcolemma begins to extend and invaginate into the interior of the cell, and these are called T-tubules, right? So these T-tubules make up something called the muscle triad, okay, or a triad in our muscle fiber. And triad is referring to three of something. And so what you have is you have two regions of the sarcoplasm, of the sarcoplasmic reticulum, and then you have one T-tubule, right? So these kind of large swollen ends of the sarcoplasmic reticulum are called the terminal cisterna, or cisternae if you're talking about multiple, the terminal cisternae of the sarcoplasmic reticulum. So all of this blue network here, this is all sarcoplasmic reticulum, but this large swelling here, that's specifically called the terminal cisternae of the sarcoplasmic reticulum. So you have one on one side, one on the other side, and then in the middle, sandwiched between the two, you have a T-tubule. So these three structures here make up what is called the muscle triad or the muscle fiber triad, all right? So we see the things that are happening along the sarcolemma. It's essentially the same thing that's happening at our neuron, okay? So we have some resting potential, and we said earlier that it's a little bit more negative, so it's about -90. But then the process and the steps to get there are basically the same as the neuron. So you have to go up and reach threshold. Depending on what source you read, it says it's -65, -75 or so. You have to get up. You move up in depolarization. Sodium is rushing in because voltage-gated sodium channel is opened. You then get to some peak. Voltage-gated sodium channel is closed. Voltage-gated potassium channel is open. Potassium begins to rush out. You drop the membrane potential all the way back down, and you then have hyperpolarization, and then you return back to resting potential until you get another stimulus that gets you up to threshold, and then you can have the process start again, okay? So if we take this now, we'll see that after we take our membrane potential, we relay our action potential from the neuron to the muscle fiber, travels along the sarcolemma down the T-tubule, and as it travels down the T-tubule, we stimulate the release of calcium from the sarcoplasmic reticulum at that triad that we just mentioned. Calcium diffuses into the sarcoplasm, and then we get into the actual mechanics of the muscle contraction, right? And this is known as the sliding filament model or the sliding filament theory of muscle movement, okay? So this is essentially saying this is the best explanation. This is the process by which the mechanism for muscle contraction is explained, right? So we'll look at these and see what happens at each phase, okay? So first you have calcium bind to troponin, okay? So calcium was in the sarcoplasmic reticulum. You had the action potential rush by, open those voltage-gated calcium channels, and calcium rushes out, right? Calcium rushes out of the cell, or I'm sorry, out of the sarcoplasmic reticulum into the sarcoplasm, and then it finds its way and it binds to troponin, okay? There's an active site and a binding site on troponin that allows the calcium to bind. You can think of calcium as the key and troponin and tropomyosin as the lock to access the myosin head binding sites on actin, right? So if you notice, you initially may have some binding sites occluded, they're covered, but then whenever you bind calcium to troponin, you now expose them, right? You now have these open regions where the myosin head is capable of binding one, okay? It turns out the myosin head doesn't automatically bind it, but it can, okay? But it doesn't automatically do it, right? So upon ATP arriving at the myosin head, so there's a binding site for an ATP molecule on the myosin head, the ATP is split, all right? So it's hydrolyzed into ADP, so adenosine diphosphate, and just phosphate. So ADP with 2 phosphates and just the 1 phosphate make adenosine triphosphate, or 3 phosphates, right? So when you do that, when you split the ATP, you release a little bit of energy. That energy release is enough to swing the myosin head over to where it forms a connection with the actin filament, right? So here you see it's not connected. You split the ATP into ADP and P and phosphate, and it swings over, right? So this is what said it's in the cocked formation. Now this process of swinging the myosin head over is known as cross bridge formation, right? We're forming a cross bridge between the myosin molecule and the actin molecule, okay? So you then have the head of the myosin pivot whenever you lose the phosphate, okay? So the phosphate comes off, that releases a little bit of energy, and it powers the myosin head to swing, to pivot, and when it does so, it swings the and pulls the actin over the top of itself. So the myosin head pivots and it pulls the actin over the top of the myosin. So it's really the actins that are moving, and it's just the myosin head that's pivoting to allow it to move. Okay, so this process, whenever you have your myosin head in this position, it's in this cocked position, and then it swings and it pulls the actin, this is called the power stroke. Okay, some books you'll see it called the working stroke, but I like the term power stroke more. Okay, all right, so the head is going to remain attached to the actin until you have a new ATP molecule come over and bind it, right? So here the head is still attached. We have a new ATP come and bind it, and it detaches. So here it's attached, and number 5 is detached. So we've broken this cross bridge, and now we're back at step 1, right? So step 5 and step 1 are exactly the same. You can wait for more calcium. You can split the ATP. You can have the cross bridge formation, power stroke, and all of those events of the contraction. Okay, so you can see in this animation, you can see exactly what's happening. So calcium has bound. We shift the troponin tropomyosin. Here you'll see the binding sites are now exposed. The myosin head binds, and whenever it pivots, it slides the actin over the top just like that, right? Now, the only thing that I don't like about this animation is that it does not show ATP coming in and being hydrolyzed, right? So here you see the binding sites are exposed. Before the heads do what they just did, before they swing over, you have to have ATP. And then before they pivot like they did just there, you also have to have ATP as well. So this is a good animation. It shows it shows the cross bridge formation, it shows troponin, tropomyosin, and calcium, it shows the power stroke, but it does not show the ATP, the role of ATP. And so keep that in mind as you're watching this, is that in reality, it's really missing a step. Okay, so here you can kind of see some of these steps kind of outlined. So you have release of, and these are numbered 1 through 9, and they correspond to the images, but you see the action potential travel along the sarcolemma down the T-tubule, release calcium. Calcium binds to troponin. Troponin is able to form a cross bridge. ATP is split. The myosin head will pivot and perform the power stroke, and then it will remain bound until you have a new ATP molecule come in and bind to it. And then you have all the different events that are involved in muscle relaxation, right? So you have to have acetylcholinesterase break down acetylcholine at the neuromuscular junction. You have to have calcium be reabsorbed back into the sarcoplasmic reticulum. It goes back into the sarcoplasmic reticulum via active transport. And so the active pumping of calcium back in, you recover the active sites for the myosin head on the actin, and this prevents additional contraction from occurring. Okay, so that is the kind of sequence of the muscle contraction. It's a lot of different steps, a lot of moving parts. So I encourage you to approach it in those 3 different phases that we talked about. So first phase, what's happening at the neuromuscular junction. Second phase, what is happening at the sarcolemma, T-tubule, and sarcoplasmic reticulum. And then phase 3, what is happening at the level of the myofilaments. So between actin, myosin, troponin, tropomyosin, calcium, what's happening there. So I would approach it in those 3 phases, and that may make learning it a bit easier. Okay, so we will stop here, and we'll pick up with our 4th part in the next video.